BJ is one of the foremost people in terms of trying to put together brain imaging, now recently adding genetics, in an attempt to understand the teenage brain. And since all of you went through that phase of life, I'm sure that you can understand that trying to understand a teenager is not straightforward. So BJ got a bachelor's degree at Appalachian State University, and that was in 1982. 2 years later, she got a master's degree. And then in 1990, she got a PhD degree from the University of South Carolina. She then went to the NIMH, National Institutes of Mental Health, as a postdoctoral fellow, and for 2 years as a staff fellow. She then did a stint as an assistant professor at the University of Pittsburgh, moving up to associate professor in the Weill Medical College at Cornell, and then professor, where she is now, and she's the director of the Sackler Institute in Psychobiology. She is also a member of the MacArthur Law and Neuroscience Network, and her work has just caught the attention of people all over the world. It's been featured on PBS, NPR, the New York Times, the Wall Street Journal, USA Today, Time Magazine, on and on and on. Because I think everybody understands that trying to understand the teenage brain is an important, difficult, but essential thing for scientists to do. In an effort to shed light on this topic, BJ has published 124 peer-reviewed papers. She's a member of 9 distinguished scientific societies. She's received 15 prestigious awards. It goes on and on and on. She's even participated in training judges. And in 2013, I think I have the year right, she participated in a symposium for the Supreme Court judges of Washington State, and is to try to teach them something about neuroscience and the law. So it's another example that I face about once a month when I stand here before you, feeling totally humbled and daunted by the person that I'm introducing. And so with that, I give you BJ Casey.
Thank you. That was an incredible introduction, and I think the only place I can go from there is down with that high expectation. But I just want to say today's been wonderful. I grew up in the South, and it was like Southern hospitality with Tara escorting me everywhere, and she's going to be escorting me to the airport too. And then meeting with the students, CP and Princess. It was a fabulous lunch. And then also to be able to have time with, a little bit of time with Mike and also Brad to talk about some of their relevant neuroscience. And look, I'll have to admit, the most important slide I'll present today is this slide, and I'll end with it as well. It is the case that every single interaction, discussion, experiment, chance meeting that we have significantly influence our thinking and significantly influence our work. So I want to sort of give you this journey that's occurring in terms of trying to understand the adolescent brain, and to really underscore this point about how it's really become somewhat of a media darling. Everybody wants to cover it, and in part that's because now we have the ability to voyeuristically, as adults, look under the hood of the behaving, acting teenager, and also because of some of the interesting behaviors and activities that they get involved with and get such jazz from. So it's probably good if I start by defining what we're referring to as adolescence first, and then we'll go from there. So we're basically talking about a period during which an individual is transitioning from dependence on their parents to relative independence. And we typically talk about its onset at around the onset of puberty. It's offset if they're really independent from their parents. May go on indefinitely financially, for many years. But it's really the point into which they can begin to use the rules they've learned about society and be a pro-social participating adult. And the evidence that I'll present today will provide a few examples of how we're seeing in the laboratory, as well as we've all seen in the real world, how adolescents engage in more experimentation. There's a heightened sensitivity to rewards, emotions, threats, and particularly social influences. And we think it's this heightened sensitivity that leads then to some of the novelty seeking, as well as the risk taking that they engage in. And it's not special to humans. Adolescence is observed in many mammals, and there's a tremendous amount of rich body of work in rodents. And I'm going to end the talk with some of that work because I think it's quite relevant in terms of thinking about human behavior. Now, every time I present these statistics, I get a visceral response because my son is still 19, left home at 17, but it is the case that we get this 200% increase in mortality rate during this period, and it's not due to disease. It's all preventable causes if we just knew how to prevent them. And then it's also a time in which there's experimentation, there's peaks in the first experimentation with substances, as well as we all know about this inflection in criminal behavior, which I'll come back to at the end when we hopefully will have more time for Q&A and discussion. This inflection I'll keep showing you throughout. This is adolescent specific. Here's another one, first use of a substance, adolescent specific. So really the overarching question that we've had is, we're seeing the changes in behavior. What are the underlying changes in the brain that are occurring during this period of time? And how is that specific to adolescence? Not just different from the adult, because if it's an adolescent phenomena, it's going to be different from children too. And so I'll be highlighting that as well. We know from postmortem human studies, as well as a number of animal studies, that there are significant changes throughout the brain in myelination, that white matter that helps neurons communicate with each other more efficiently. We also know that there are regional changes in the brain in terms of the number of synapses, these points of connection between neurons, and that these changes are really occurring in environment and experiences. I used to think I could lock up my son until he was 21 and then everything would be fine and let him out. It's not true. I want to just start by saying maturation is not magical. It is occurring, and these circuits are getting wired in the context of experiences. So it's very important for us to think about the right experiences for them to have to master this transition into adult roles. And that's particularly interesting, of interest when we start talking about the juvenile justice system. Now, the most overplayed movie in all of developmental neuroscience, I think, is this one. And I used to be a postdoc at the NIMH in Judy Rappaport's branch, where Gautier and Guide both worked. And basically, what this movie is showing you is those areas that are blue are areas that are reaching cortical thickness, the adult peak levels of cortical thickness. And if I play this again, what you'll see is there's some areas that are reaching that thickness sooner than others. And I think it's a lot easier to see on the static picture. So let's just look at these bottom pictures. This is as if we're looking down on top of the brain. And this is the motor strip. This is the back of the brain, the visual cortex. It really makes logical sense that systems that help us see, like the primary visual centers, and help us move and act in the motor cortex, would fine-tune and mature before those areas that use those systems to plan future behavior would develop. And so that's what we're seeing on the cortical surface. But when we talk about the adolescent, it's not so much about the cortex when we really think about the behaviors that they're engaging in. It's more these primitive, deep structures in the brain that are associated with rage and desire and fight and flight. And this is an old slide from Terry Jernigan's lab when Elizabeth Sowell was training with her at UCSD. And it's basically showing in the front of the brain these fuchsia blobs are the prefrontal cortex, but then you have these deep structures as well that are continuing to be modified and change as the circuit begins to form with experience and maturation, and these regions are talking to one another. All these regions are there early on. It's just the way that they're, over the course of development and time, they begin to talk to each other differently. And so I think this movie kind of captures it. The areas in red that you see in this glass brain are those deep emotional centers in the brain, and they're changing in many ways during the adolescent period and well before this area in green. That's the prefrontal cortex that's so important in rational behavior and self-regulation before they're fully developed and there's mature connections. So it's harder earlier on for them to sort of quiet a system that may be activated during a time when the brain's being marinated in gonadal hormones. It's a time when we see peak changes in growth factors that are so important for learning and development, and also a time in which neurochemicals are regionally changing, just as I showed the cortical surface was changing. But we think this is resulting in a tension between these two systems, in which then leads to this imbalance model that Adriana Galvan, a now tenured faculty at UCLA, kind of pulled together when she was a doctoral student in my lab. And this is showing that if you have areas that have a slower development prior to those that might develop earlier, then there's going to be a tension or imbalance when both systems are called upon that you don't see when they're both maturing or when they're both fully mature. There are a number of other names for this, a dual systems model. We think of it more in terms of circuit, and then there's also a triadic model that's been proposed. But for the remainder of the talk, what I want you to focus on is really when you start to see patterns like this, that are either an inflection or else you get a deflection in behavior and in patterns in the brain. We're also very interested in adolescent emergent and also linear changes that could be decreasing or increasing. But this is where we began to see the same pattern as what I showed you with regard to the inflection in criminal activity, also substance use, exploration, and novelty seeking. And so that last part, in terms of how I defined adolescence, we'll be focusing on with regard to this heightened sensitivity reward, evidence for that, to threat, how adolescents behave differently from children and adults when presented with these cues, and how they can lead to riskier behavior. And then we'll end with implications for juvenile justice system. That I've learned some great things are happening here in Madison. It's always wonderful to hear states that are doing so much for young people. So I think it's probably most obvious if I start with money. Everybody has an appreciation for the rewarding qualities of money. And so Adriana Galvan, who developed that imbalance model, one of her first experiments was just to see how the representation of money and reward was different across different ages. And so she used a task that really built on paradigms that had been used in non-human primates to understand reward circuitry, particularly the dopamine system that's so important in learning about positive outcomes and motivated behavior. And so basically, she came up with a task. Here, I'm just going to back up here. With 3 discrete stimuli, we call this the pirate task, and each of those stimuli, if you correctly responded to what side of a screen they were presented on, you got one of 3 different payouts. So one was always associated with small, one with medium amounts, and one with a lot. We didn't use actual dollar amounts because the adolescents started to calculate how much they were earning, and it became an arithmetic task. So we started doing everything visual for them. It turns out, and other laboratories have shown this, over a period of time, you begin to realize what cues are associated with what outcome, even if you're not consciously aware. And in this experiment, none of the participants could tell us which pirate was associated with what exact value. Some of them thought there was an association, but they couldn't articulate it. And here's what the data looked like. So this is how fast they are, and this is over time. And so the first thing that you might expect are any stimuli that might be associated with a lot of money are going to get faster, and that's what we observed. It was actually quite interesting to see that if you only got a single coin, you're getting some money, right? But they got a lot slower, significantly slower, if they weren't getting the highest payout. It's relative, and we know a lot about that relativity of reward. Now then, if you take this task in the scanner, the whole bloody brain can light up. But what you're interested in is what areas of the brain are specifically mapping on to this change in the magnitude of reward that's been associated with the cue. And there are two regions that we see there in the reward circuitry. Again, I was talking about this tension or balance in the prefrontal cortex and in subcortical regions. This is an area in the nucleus accumbens that's very rich in dopamine and very important, learning about cues that signal positive outcomes. And also this area in the prefrontal cortex that helps you maintain that information too over time. Now, there's nothing interesting about these patterns in terms of development because I haven't broken it down by age. But now let's just look at how the pattern in the prefrontal cortex looks by age group, because this is just by the magnitude of the reward across all ages. When we now break it down and we look in the prefrontal cortex, we see something that is almost close to linear or almost emergent, but it's not an inflection in any way. Where we see the biggest bang for our buck in terms of that inflection is this period of time during adolescence, so that when they're getting a large reward, they're activating this region of the nucleus accumbens much more than children or adults are. And what's interesting about this in particular is it's this region of the nucleus accumbens that's been associated with just before you're about to make a risky decision, you see activity in this region. And when we asked our participants the likelihood of them engaging in risky behaviors that you can ask across all ages, such as, what's the likelihood of you leaving a party with a stranger? Or what's the likelihood of you engaging in risk-taking type sports like rock climbing and the sort? What we see is the amount of activity in this region to large monetary reward is associated with the likelihood of risk-taking behaviors, regardless of age. So when you get a bump like that, it looks like we're seeing an increase that's actually mapping onto real-world behavior. But often, when we talk about adolescence, we talk about self-control. And I want to just digress for a moment and distinguish between a sort of simplistic view, and I apologize to some of my neuroscience colleagues in the audience, or hot-cold versions of impulse control. So a hot one is when you're making an impulsive response in the heat of the moment. There's something emotional or money, positive or negative things involved. Cold impulse control is more like when you get into a habit of doing things. So if you have a habit of walking to a certain building every single day and there's a day where you have to do something different or go a different route, sometimes you find yourself still going to the same place and then you have to correct it. So how you can measure this cold version, and then we'll come back to the hot version of impulse control, is to use neuropsychological tasks like the go, no-go, where you build up this tendency for the participant to want to respond because you have these frequent targets that they're going to. And then there's this rare cue that pops up where they know they're not supposed to respond. So that everybody, it's the afternoon and it's sunny, thank you for being here, so why don't we have some participation? And basically, anytime you see this little tricky mold that's in the garden, just say whack. And this is relevant in Wisconsin, not so much in New York. But don't whack to the eggplant that's magically appearing from the ground. New Yorkers don't know about root vegetables coming out of the ground. So, but that's not what you want to whack, is the magical eggplant emerging from the ground. So with these tasks, you go really quickly. So just say whack whenever you see the mole. You ready? You're first going to see the hole. Oh, this is going to be a good group. Okay. All right, so go as quickly as you can. You ready, set, go. I love it when people catch themselves. And no is, that was pretty tame. Thank you, since this is being recorded. So when you perform this task, what's really interesting is there's a lot of individual variability regardless of age. But there's this general tendency for us to improve with age. And this is just plotting D prime. It's simply just incorporating when you accurately detect what you're supposed to be detecting and push the button, as well as when you're supposed to be stopping, but yet you can't stop yourself. So it looks at misses and these false alarms. And we should note, on cold tasks like this, it's sort of like a habitual response that you're making. There are adolescents who perform better than some adults. So there is a lot of individual variability. But in developmental studies, I think this is where the sort of meat and potatoes are, as someone from Ireland would say. But if we can put them in these neat boxes, and then you begin to see these developmental changes. Nothing special here in terms of adolescence, right? This looks adolescent nonspecific. It looks like there's a general tendency to improve from childhood to adolescence to adulthood. And if you look at what brain region is correlating with how well you can stop yourself and, or how poorly you're doing, it's an area in the prefrontal cortex. One that I'm sure Kent Kill talked about a bit yesterday in his talk. So that the activation when you correctly inhibit the trial is actually correlated with how well you perform the task overall. And this is just showing you there's a lot of variability in individuals and also across development. But now, let's make this impulse control a bit more salient and turn it to a hot version. And that is by simply asking, what's the impact of positive information, such as social cues? We could have looked at cues that have been associated with reward, but during adolescence, there's so much of reading into facial expressions and trying to understand where they are in their peers as they're forming their self-identity. So I want to highlight some findings that came from Leah Somerville, who did her undergraduate work here and actually worked with Ned Kalin before going to graduate school at Dartmouth and doing a postdoc with me. She's now a faculty member at Harvard, so Madison can take quite great pride in her work. And what Leah did was she examined how well across development you could stop yourself from responding when it was a positive social cue as opposed to neutral information, and if that varied as a function of age. And here's what she found. Adolescents are pretty bad. When you tell them, don't go there, when they see a positive social peer, it's like they can't stop themselves. These are rare. Remember, they already have this tendency to respond, and that seems to pull them over. And that's quite different from what we see in children and adults who make an equal number of these errors to neutral and positive information. And then if we look in the brain to see what's the area that's coupled with this pattern of brain activity, we actually see this region, again, in the nucleus accumbens that I showed you before, is activated when you're getting large amounts of money and is also associated with risk-taking performance. And positive social cues, it's one way to look at it in the laboratory, but we know that just the presence of a peer can impact adolescents' behavior, can actually impact ours as well. But I wanted to just highlight Larry Steinberg's work. Many of you may be familiar with some of the work that he's done by looking at decision-making in adolescents and adults when they're performing decision tasks with a peer present as opposed to by themselves. And he has this nice sort of experiment that we've all found ourselves in as we drive, and it's basically just seeing how risky their decisions are when they come to a yellow light. It doesn't mean drive faster and get through, right? We have to judge the distance and the timing before we make a decision. And adolescents and adults, he had younger and older adults as well. So there were 3 groups, and he just looked to see how they performed this task in terms of risky decisions, and also if they didn't make it through the intersection, then you saw the crashed windshield, and that was a crash. And so here are those data. If you look first just at these blue bars, these are risky decisions and crashes when they're alone, and there's no difference between the developmental groups. The only time you see a difference in their risky decisions and in crashes is when a peer is present. Now, he recently scanned this task, or scanned individuals while they were performing this task, and their peers were right outside the scanner room. And he saw this performance. He saw even bigger performance when they were right next to them, but testing them behaviorally. But when you look in the scanner, it's that same area in the accumbens when they make a risky decision when the peer is present that is activated. Again, more evidence about this circuitry that's very important in terms of engaging in risk-taking behaviors. But so far I've been talking about sort of positive social cues and peers and rewards. But what about negative information? Adolescence has been described as a time in which there's this heightened emotional reactivity, and often we think about that as the storm and stress or negative emotions, about there's no tomorrow and things are never going to be right. So we were very interested in just how well an individual could control their impulses in the presence of threat or negative information. And so we used cues that there might be a biological disposition, but really over a lifetime, we come to associate an expression like that with somewhere, someone, something is going on. And so we stop and we orient to it, and then we determine whether or not it means anything in the situation that we're in. So this is work that was done by Todd Hare, who went on to do a postdoc at Caltech and is now at Zurich as a faculty member, and an MD PhD student who's just joined the lab and reanalyzed some of that data, Michael Dreyfus. And so it's almost identical to what you saw before. It's that same whack-the-mole task, except we're using facial expressions, but now we're looking at how impulsive they are when there's a threat cue as opposed relative to a neutral expression. And we see the same pattern, as if they're drawn to danger, so that they're making many more commission errors than what you're seeing in children particularly, but it's also different from adults. And I should also just note that if you break this up by males and females, the males are predominantly driving the effect, but it's significant across the group. You really need a larger sample, and many of these imaging studies haven't been quite large enough to give you the power to do those more complex interactions. But what we also know about these threat cues is the area I've been talking about before is the nucleus accumbens, but another area that's very important in determining the emotional significance and outcomes of information in the environment is an area called the amygdala. And the amygdala shows a heightened response in adolescence relative to children and adults too. But what's really important, and Mike and I were actually talking about this before, is maybe not that initial response, right? Because if you think there's threat out there, it's adaptive for you to have a response, right? You want to make sure that you're not taken out by a predator. It's something that we've evolved to be adaptive for survival. But what's really important is whether or not you can regulate that and bring it down, and how important it is for our teens to learn how to do that for themselves, because we as parents aren't going with them to their social gatherings once they become teenagers. Sometimes we do, but it's not their preferred manner of attending a party. So basically, what we see is that the ability to bring that system down, or for it to habituate over time and recognize in the current context there's safety, is this area in the ventral prefrontal cortex that projects to inhibitory cells in the amygdala as worked out in the rodent system that can shut that system down. And so basically, the more activity that you have in this region, the greater the amount of decrease in the signal in that region from early trials to late trials. And I want to show that to you in a movie, because I think sometimes a movie tells 1,000 words. And it's also this ability to regulate your emotion when you can't, that you begin to get into pathological states of anxiety. And that's very important in terms of the adolescence, because we see peak prevalence of anxiety in early adolescence, even though it emerges a bit earlier. So here's just a movie of the typical response in an adult versus an adolescent when they're seeing these cues of threat pop up on a screen. And you're looking into the brain at these deep structures in the amygdala. And so what you see is initially there's this heightened response with these cues, and then with repeated exposures, it's like, all right already, nothing's happened to me in this context. This means this is not a threat cue for me. And it goes to baseline as indicated in the blue activity that you're seeing. Now, This is in sharp contrast to individuals with anxiety and also in many of our adolescents. And here's what you see. You get bilateral activation in the same region, and even with repeated trials, we're still seeing activity in this region. Now, one of the concerns that we've had in terms of using facial expressions like these with adolescents is exactly that bias I told you before, and that is they can read emotion into any expression almost, or they seem to. You can just be thinking about something else, and your teenager will think a peer is thinking something badly about them when they're not thinking anything at all. And then they think the world's going to end, and then they can't think about the future, and everything's going to be all right the next day. So we've been interested in whether or not these threat cues really have significance, and is there a way to take neutral information and make it be associated with something threatening, and then see if there's a developmental difference, or is it just specific to social cues? And the way that we've done that is to actually turn to doing parallel studies in mice and humans where we can look at fear. And this work is in close collaboration with Siobhan Patwell, who is a graduate student, with my close collaborator, Francis Lee, with whom I've done a lot of genetic work, but I won't be talking about the genetics today. And so basically, what we can do in the rodent work, and particularly the circuitry that we worked out on threat and fear, is you take a neutral stimulus and you pair that with something bad, such as a little foot shock, and it doesn't take too many pairings of that tone with that foot shock before the animal starts to freeze just to the tone alone. And then you can look at how well they can learn when that tone no longer is associated with something bad happening by putting them in a new context and just playing that tone over and over again and see how well they learn. And that's extinction. Something else that you can do is not just look at cues, but another area that we're investigating are contexts, because there's situations that are very important for adolescents too. And the same thing is true. When you want to see how well they generalize contextual fear, if you shock them in this context and then you put them in the same cage the next day where they were shocked previously, and you just leave them in there and you see how much freezing there is in that cage relative to animals that weren't conditioned. And I want to just first focus on the cued fear conditioning. Again, pairing the tones, and what we're interested in is when you're just playing the tone in a new context all by itself. Now, I mentioned that we're doing parallel studies in humans as well as mice, and we're not giving them a little foot shock. Instead, we play an aversive sound that we published on where we have aversive ratings, and it also gives you a pretty reliable skin conductance response, or where you can measure really just changes in sweat just on the fingertips. And what we do is we pair that aversive sound with neutral cues, and it can be either the yellow or the blue, and you want to have two, so you can always compare the difference between the two to determine how well they're learning something. And then during the extinction trials, you see how much of a skin conductance response are they showing to the one that was paired relative to the one that wasn't. And first, I just want to show you that there is all ages can fear condition. They can all learn an association pretty quickly, and this is just showing you children, adolescents, and adults are all having more of a skin conductance response to the square that was paired with the aversive sound as opposed to relative to the one that was not. But then when we look at the extinction data, it's pretty interesting, and it's a similar pattern in the mice and the humans. And now I'm presenting the higher the bar graph, the better the learning, and that means the bigger the change. And so the pre-adolescent mice and the children are showing tons of extinction, and the adults are showing extinction too. The adolescents still seem to be showing that arousal response as measured by skin conductance in the human, and as measured by freezing behavior in the mice. And it turns out that if you test these mice later, they still show fear to this particular tone. So that sort of goes with this notion of when I was showing you the threat cues that were popping up, it suggests that it's not simply associated with social cues that might be negative. But it's actually cues that are associated with threat that they're learning, where everybody coming into the experiment, every age group, had the same amount of experience with that pairing. So now I just want to turn to the contextual fear learning. And so again, this is, they get a foot shock one day, and then you bring them back and you put them back in the same cage that they got the foot shock. And what we see here is that the adolescent mice look fearless. Pre-adolescent mice and adults are showing freezing. And the adolescent mice, you put them back in the cage, and they look like they don't care. They absolutely look like they don't care. Does that mean that they can't learn to care or learn about fear at this time? And I'll have to tell you, it is always not what you predict, and that sort of serendipitous finding, because, you know, if we knew everything there was to know about science, we'd prove all our hypotheses. But that's not the way it is. We have these huge paradigm shifts in our thinking when something unusual happens. Well, if you've ever done developmental studies or animal work, it's hours of time watching the behavior of the subject, either the animal or the individual, that you're recording when they're freezing or not. So Siobhan Patwell, who was a graduate student, was trying to figure out an automated way to measure freezing behavior without having to spend so many hours in the lab. And so basically she, I guess this was about 1 to 2 months later after she had done this experiment in the contextual fear learning. And she's trying out this new software, and she just takes a random cage of animals and she brings them into the testing room. And she puts the first animal in, this is an adult animal, and it jumps out of the cage. She goes, that's weird. She puts him back, and she takes another animal from that same cage, puts him in, and he's just huddled over in a corner like he's really afraid. And so she thinks, somebody didn't clean out this cage. You know, it must have the odors of other animals that have experienced it. And so she cleaned it out, made sure, and she put those same animals back in, same response. So she went, something's really funny about these animals. She went back and she looked at all the recorded notes on the cage about what experiments these animals had been in. And these were exactly the animals who showed no fear as an adolescent, but as an adult are showing the fear. So it's not that they're not learning, they're just not expressing that fear. And this is really important when we think about adolescent behavior and when they appear fearless, but what their ability as an adult is and what they do remember from their adolescence, even if they're not showing it to us. And I think this is very important in terms of some of the issues with juvenile justice too. But when you see behavior like this is typical behavior. What I've been showing you is all data from typically developing animals and human animals. And earlier I was showing you typical brain development. So the big question is, why would the brain be programmed this way to make us so fearless of new contexts, but so vigilant of cues in the environment, and so pulled by rewards and positive social cues? So I always like to sort of begin the conclusions by us thinking about when things would be adaptive. So let's think about the life of the adolescent. It's pretty good, for the most part, in terms of they have shelter and they're getting some sources of food. But if they continue to stay in that shelter and eat all the food, they're going to deplete all the resources. So they need to be pulled out to begin to explore new sources of food and also to explore new possibilities for mates so they can successfully procreate. But if you're pulled out into the environment and a predator gets you, you're no better off having gone out there than staying home, right? So it's important to be vigilant about cues that may predict danger and to be vigilant about those. However, if it's not cues, but it's context, and we know every situation we go into, it's not like everything's hunky-dory, every new situation we're in, every new talk that we give. So it is probably adaptive for us to not be afraid of new contexts when something bad happens, or we might not ever want to leave the nest. And then finally, why would you then have this delayed memory of fear? Well, it would make a lot of sense, and it's right about the time, I didn't point to those ages, just at the time when they become capable of reproducing. So they're already in early adolescence, but when you become capable of reproducing, that's when they show the retrieval of that fear memory. That's when you're settling in a niche, and you don't want to have your nest in an area that you remember as being dangerous. So let's move from this and end, hopefully, with as much of a discussion in terms of the implications for policy. And I think there are a couple in terms of diminished culpability and also in terms of the way that we're thinking about rehabilitating individuals to reduce recidivism and help them get through this period, this typical development mental stage where they have to learn how to negotiate the world on their own at a time when they're having physical changes, their sexual changes, their more cognitive demands on them and the sort. But there have been at least 3, right, at the level of Supreme Court. And now we've abolished the death penalty, and the most recent decision in 2012 was actually abolishing mandatory life without parole, although that doesn't mean that an individual isn't necessarily given that sentence. It just means it can't be mandatory. These last 2 cases, Miller and Jackson, were really interesting in the context of the examples I presented to you. In both cases, they were 14-year-old adult males. In both cases, they had peers or accomplices. Both cases, there was a homicide. And in both cases, there was an emotional setting or situation in which these crimes occurred. So as a group, we can talk about policy change with regard to, it's not that adolescents can't make decisions. When I showed you just straight impulsivity, doing whack the mole, there's some adolescents who can perform that task as well as adults. But in the heat of the moment, it's hard or difficult to stop an action. And many times you'll hear teenagers say, I knew better, I just didn't, I don't even know why I did it. And I think that's very important for us to keep in mind, because it also comes into play in terms of policies and when teenagers can make decisions about their body or about who they want to live with. And I think it's very important that we bring all of that science to the table and thinking about all levels. The question is, to what extent can we apply the data that I showed you today to individuals? This is all correlational. I tried to show you the individual variability that we're seeing across these groups, and that's very important to remember. And so when I'm talking to judges or people working in the trenches, basically when they say, well, how does this relate to the individual? I just have to say that at present, we really don't have the support to, the evidence to support such an inference to the individual. This is almost all the work I showed you today is based on group data. And then I mentioned that as a group they have these tendencies, but this difficulty seems to be this tension or imbalance as we're seeing changes in local circuitry in these emotional centers, primitive centers in the brain, before the more, as Jonathan Cohen would call it, vulcanized prefrontal cortex, Mr. Spock, can help regulate, the Captain Kirk passion regions and sort of regulate those. So again, even if a teen knows better, sometimes in the heat of the moment, he'll make a bad decision. And there's a sort of a discussion that Larry Steinberg and I have that's interesting about adolescence, and that is, why is it that when a baby can't walk at 2 months, we call that development, but when an adolescent can't make a good judgment in the heat of the moment, we call that aberrant or deviant? And I think we really need to think carefully about that in terms of what we've seen as the group data in behavior and in the brain. And then it's also important for us to recognize that this is transient. There's a ton that's going on during this period of adolescence into young adulthood. And I wanted to just end on this point, and that is, how does this inform rehabilitation? We know that punishing behavior doesn't change it. We know positive reinforcement does have a bigger influence on changing behavior. And so we need to think about opportunities in which we can avoid recidivism and use the empirical knowledge that we know, and also what the actual goals are during the adolescent period. So they are trying to become a pro-social adult. But if they don't have pro-social role models and they don't have opportunities, the circuitry is not going to mature magically. They have to have opportunities where they're regulating their own behavior in the face of conflict. And they need opportunities to do this, and they need opportunities to engage in pro-social behavior, and they also have opportunities for pro-social roles. They do need to be held accountable for their behavior. But if we're going to help them and prevent them from coming back in the system, then we need to really work with our goals of adolescence. So what I'm going to do is to stop here and really just remind you that it's not all thrills during adolescence. There are true threats there. And this is an adaptive period. And it's an adaptive period that's helped the species survive. And it also goes along with the fittest of the species ultimately surviving too. So thank you very much for your attention.